Hello and welcome to this Revision Monkey video on Biology Paper 1 required practicals. The first 5 are for combined and separate science, and the 6th practical is a separate science only practical. Microscopes required practical. With the microscopes required practical, you need to know how to prepare a slide and how to focus that image under a microscope at different magnifications. So first of all, in the microscope, you've got the bit that you look into, and that is called the eyepiece. That often has a number on it, for example, times by 10. We'll come back to that later. You've got the objective lenses here, and often they have different numbers on. For example, you might have times 4, 10, and 40, for example. Now, to work out what magnification you're using, you multiply the objective lens by the eyepiece number. For example, if you looked at 4, that would be 4 times 10, which would be 40 times magnified. The 10 lens would be 10 times 10, which would be 100 times magnified, and the 41, 40 times 10, which is 400 times magnified. This here is the stage where you put your slide that you want to view. And often on the arm here you might have 1 or perhaps 2 focusing wheels. The larger one will be for the coarse focus, so you can just call that a coarse focus wheel, and the other one would be for smaller adjustments, which is the fine focus wheel. So now I'm going to take you through the steps to carry out this required practical. First of all, to prepare the slide, you need to spread the cells on the slide, add a dye, for example iodine, to stain the cells, and then add a cover slip on top, and this is just a small transparent square so that you don't get any of the dye onto your lenses. Place the slide on the stage and use the grips to keep it in place. Select first the lowest magnification objective lens to view your cells. So for example, in this case, we'd make sure we start off with 4 times 10, which is 40 times magnified. We use the coarse focus wheel and then the fine focus wheel to focus the image. And we would then draw the image using a pencil and no shading of any of the subcellular parts. So for example, if we were drawing an animal cell, we might draw the cell like so. With the nucleus, we wouldn't shade it in, but we'd make sure we labelled the bits that we saw, for example, the nucleus and the cell membrane. Finally then, we would then change the lens to a higher magnification and refocus the image. As an extension to this required practical, you might be asked to do a magnification calculation, for which you need to remember this equation. Magnification equals image size over real size. For example, a bacterial cell was viewed at 100 times magnification under a microscope. The width of the bacteria cell in the photograph, over here, is 20 millimeters. What is the real width of the bacterial cell? Give your answer in micrometers. So lots to do here. First of all, we need to work out what we've got. We've got a magnification, and we've got the size of a photograph, which is an image. So this is our image size, and we need to calculate our real size. So first we'd rearrange this equation. So it would be real equals image over magnification, and then we put our numbers in. The real size of the bacterial cell will equal 20 divided by 100. And this will give our answer in millimeters. So our answer is 0.2 millimeters. Now we need to change our answer to micrometers, and to do that we times by 1,000. So our final answer would be 200 micrometers. Osmosis required practical. So let's start by reminding ourselves of the definition of osmosis. Osmosis is the movement of water from a dilute solution to a concentrated solution through a partially permeable membrane. Now you may well know a definition that talks about the water moving from where there is a high concentration of water to where there is a low concentration of water. And it means the same thing. A solution with a high concentration of water is called a dilute solution, and a solution with a low concentration of water is called a concentrated solution. But you're more likely to see the words dilute solution and concentrated solution in an exam. So we're talking about the movement of water, and we're talking about the movement of water in and out of pieces of vegetable. Now this could be any vegetable, for example potato or carrot or beetroot, anything like that. And the vegetable itself has a natural sugar solution inside the vegetable, so sugar and water. In the experiment you will put the vegetable pieces of approximately the same size, so you'll try and make the width and the length the same, but they're naturally going to have slightly different masses, and you account for that by looking at the change in mass, which we'll talk about later on. You're going to put these vegetable pieces in different concentrations of sugar solution. For example, here 0 moles per decimeter cubed of sugar, so no sugar in this one, just pure water. And you'll see the movement of water by osmosis from the dilute solution outside to the more concentrated solution inside. This one again, a little bit of sugar in here, so you might see the movement of water inside from a more dilute solution outside to the more concentrated solution inside. And this one here, at the high concentrations, you may well see the opposite, whereby the concentration of sugar solution inside the vegetable is more dilute, which means the water will move out of the vegetable. Now in terms of mass change, if we measured the mass of the vegetable before and after, because this gained water, you would see a mass increase, and the same for this one. And because this one lost water, you would see a mass decrease. So if I take you through the main steps of the practical now, you cut vegetable pieces into approximately the same size and shape. Using a balance, weigh each vegetable piece and record this in a table. Add one vegetable piece into, I've just suggested here, 200 ml, it doesn't matter too much as long as you keep that as a control variable, of 0 moles per decimeter cubed sugar solution. Another vegetable piece into 0.5 moles per decimeter cubed of sugar solution, and another into 200 ml of 1 mole per decimeter cubed sugar solution. Now again, these solutions you can change if you are asked to write it from scratch in the exam. You don't have to stick to these exact ones. Leave the solution and the vegetables for 24 hours. And then you're going to remove each vegetable piece, pat the outside of it dry with a piece of tissue paper or a kitchen roll, just so that any water on the outside doesn't affect the mass when you're weighing it, and then re-weigh it using a balance. So if here you've measured the mass before, and here you've measured the mass after, we can calculate whether it's got an increase in mass or a decrease in mass. And then we'll use those values to calculate the percentage change in mass for each vegetable piece. So you might get results that look a little bit like this. So we've got our different concentrations of sugar solutions here. The mass of the potato, if we use a potato for example, before. Now these are ever so slightly different. We've tried to get the size the same, but they are a gram or so out of each other. The mass of the potato after the 24 hours. And then we've calculated a percentage change in mass. Now to do this, you need to remember this equation. Percentage change equals change divided by original times 100. So if I go through the first calculation for you, you'd have found the change in mass. So 15 for the first one, for this data here. 15 minus 12 will give us the change. Divide that by the original, which was 12, and multiply that by 100, and that will give us our 25% increase in mass. Now we know it's an increase because it's gone higher just here. Now for the final mass here, you can see a negative percentage change because the mass has actually decreased when the vegetable was put in the 1 molar per decimeters cubed solution. So a negative suggests a decrease and a positive suggests an increase. And then we may well be asked to plot these on a graph. So obviously you'd have your 0 here, and going up in times tables you'd put your percentage change in mass. I'm not going to write the numbers on. The x-axis, just to make it nice and clear, they've put the words for it down here, the title here, sucrose concentration or sugar concentration, and that will be in moles per decimeters cubed. And again you'd have numbers here indicating the concentrations. So you might end up plotting some data that might look a little bit like this, to which you then put a line of best fit on your graph, going through or as close to as many of the points as possible, like so. And the next important bit the graph can show us is this intersect here, where the line of best fit intersects the x-axis. This value for sugar concentration, if you were to read it off your graph, would give you the sugar concentration inside the potato. So I will write it out here for you. This value here would give you the sugar concentration inside the vegetable that you are using. Why? Well, because at this point here there is a 0 percentage change in mass, so there's no change in mass. So what this means is if you have your potato or your vegetable inside your a beaker with your sugar solution, it must mean for there to be no change in mass that the solution outside equals the sugar solution inside. So there's no net movement of water in or out of the potato, so sugar solution inside the potato or the vegetable equals the outside solution. Enzymes required practical. For this required practical, you will investigate the effect of pH on the rate of enzyme activity. So what we do first of all, if I take you through step by step, is we would heat a water bath to 37 degrees and place a boiling tube inside containing some amylase. So I've just given some suggested amounts here, so 7 centimeters cubed, for example, of amylase, and 25 centimeter cubed of a pH buffer, and we'll start off with, for example, pH 3. So in this test tube we've got amylase going in there and also a pH buffer which will all mix together. Now a pH buffer is just a solution which has an exact pH. In this case we're starting at pH 3, so something that your science technicians could make up for you. Now we want to heat that up to 37 degrees or thereabouts. In your exam they might talk about 35 or 40, but around that kind of temperature, because this is body temperature and that is the optimum temperature for enzymes. So amylase is the enzyme we're going to look at, and we are going to then also at the same time prepare a spotting tile, which is this piece of equipment at the bottom here, by dropping a couple of drops of iodine into each well. And iodine is actually a browny orange colour. So first of all, our wells will look all orange, like so, with a couple of drops in each one. So we'll prepare that and just put it to one side. Our next step is to add 5 centimeters cubed of starch to the boiling tube and immediately start the stopwatch. So amylase, if you remember, breaks down starch, so we will add our starch into the boiling tube and immediately start our timer. Now every 30 seconds we are going to do continuous sampling. This is a method which shows continuous sampling, And every 30 seconds we are going to take a small sample of our mixture here, which contains our starch, our amylase and our pH 3 buffer first of all, and we are going to drop that on our wells. So every 30 seconds remove a small sample of the mixture in the boiling tube using a pipette and add a couple of drops to one well on the spotting tile. Now, in the first stages, our spotting tile well should go black because of the presence of starch, because starch turns iodine black. Now, this will keep going for a while, and then after a while it might go a browny kind of colour, okay, to show that there's a little bit of black in there still, but a lot less than before, maybe into a light brown. And then eventually, after a while, it will remain orange. So if we continue to sample, the wells would just remain orange, because that would suggest that the amylase has completely broken down the starch. So we would repeat the previous step until the colour of the well remains orange, and then we would stop the stopwatch and record the time. Now we can actually convert this into a rate by doing the following equation. Rate equals 1,000 divided by time, and this is the only calculation we can do when we have a colour change, because you can't have such thing as a colour change over time. So we just make the numbers a little bit simpler by doing 1,000 divided by time. So that will give us a time it takes for the enzyme amylase to break down starch, keeping the temperature constant, that's a control variable, and we've used one of our buffers, our pH 3 buffer. Now this is our independent variable that we are going to change, and our rate is our dependent variable. So the final thing we need to do is repeat the investigation, but for different pH buffers, for example 5, 8 and 13. And within the experiment, what we want to do is then finally repeat the whole thing 3 times and calculate a mean rate of reaction, which I'll just put as rate, for each pH. Food tests require practical. So there's 3 food tests you need to be aware of. In your revision guide they may talk about a test for fats as well using Sudan 3. However, because that's quite a nasty chemical, they've removed that as part of the required practical. So you shouldn't be asked on fats, you should just be asked on carbohydrates, which split into complex carbohydrates like starch and simple carbohydrates like sugar, and then protein. But first of all, you need to prepare your food sample, and you do this by crushing up your food in here using a pestle and mortar. And then you'd mix it with a little bit of water in a test tube or a boiling tube to make a food solution. So we could do 3 different tests. We could do a test for starch. So if we have our food solution in here. So the test for starch is to add iodine, and if iodine detects starch, there will be a colour change from orange to black. So if this food sample contains starch, it would turn black. So to test for sugar, we'd put some of our food solution inside a test tube, and we would add Benedict's solution. Benedict's solution is blue, but importantly we also have to put this in a water bath. So you could either heat it up and try and maintain the temperature, or you could use an electronic water bath, and it needs to be around 75 degrees C, so quite a hot water bath. And a positive result, if we put our Benedict's inside, heat it up in the water bath, a positive result would be a change from blue to orange. Now you may well see in your revision guide other colours as well, because if it's got a little bit of sugar in, it might go a green colour. If it's got a lot of sugar in, it might go orange, or maybe even a brick red colour. Okay, so in the exam they might talk about green, orange, or brick red. But if in doubt, remember blue to orange. So to test for protein, our final food test, we would add a food sample into our test tube. And the test for protein is to add Biuret solution. And that's quite a light blue solution that we add in. And that will turn to purple. Okay, it's quite a light purple, that's why I put light purple, but you still get the mark if you put purple in there as well. So if protein's present, you will get the colour change from blue to purple. Measuring the rate of photosynthesis required practical. In this practical, we submerge some pondweed in water, and the number of bubbles it gives off per minute gives us a rate of photosynthesis, and the investigation looks normally at how light intensity affects the rate of photosynthesis by moving a lamp closer or further away. For example, if you had your lamp far away pointing at the pondweed, you might get a few bubbles coming off in that minute. If you then moved it closer, you might get a few more bubbles given off in that minute. If you moved it closer again, increasing the light intensity that's reaching the pondweed, in that minute you may well get a few more bubbles giving off, indicating a higher rate of photosynthesis as you move the lamp closer to the pondweed. So let me run through the main steps of the practical that you need to get down if asked to write a method. Cut 10 centimetres of pondweed and submerge this in water in a boiling tube. Clamp the boiling tube to a clamp stand. I haven't shown this here, but you may well have set up a clamp stand just to make sure that stays in the right position, clamping it there like so. Measure 50 centimetres away from the clamp stand using a ruler and place a lamp here directed at the pondweed. Turn on a stopwatch. Record the number of bubbles produced by the pondweed in 1 minute. Repeat this experiment for the following distances: 40 centimetres, make sure you list them, 30 centimetres, 20 centimetres, 10 centimetres, each time recording the number of bubbles produced by the pondweed in 1 minute. So our variables are important here. Our independent variable is the distance between the lamp and the pondweed. The dependent variable is the number of bubbles per minute. And important control variables, things like type of pondweed, the length of pondweed, the power of the lamp you're using. For example, you might want to say you're going to keep it at a 40-watt bulb or something like that. And the temperature. Now, with regards to temperature, you may well see a transparent heat shield in front of the lamp, which will stop the lamp from heating up the water. Or you may well see that the investigation makes sure it uses an LED lamp, a light emitting diode, rather than the filament lamp, and that will reduce the heat given off and therefore help you control the temperature in the experiment. Okay, the inverse square law is for higher tier pupils only. If you were doing foundation, the relationship that you need to know is the further the lamp is away from the pondweed, the lower the light intensity. So really at a foundation level, you'd need to know as distance increases, the light intensity decreases. However, for higher tier, you need to know a very specific relationship between distance and light intensity. Yes, it's true that as distance increases, the light intensity will decrease, but it's got a very special relationship in that light intensity is proportional to 1 over distance squared. So we talk about light intensity being inversely proportional, which means 1 over the distance squared. This required practical is for separate sciences only, and it is investigating the effect of antibiotics on bacterial growth. Now, prior to this practical, the student or the technician might have prepared an uncontaminated culture of bacteria. That's when you use the aseptic technique, sterilising your inoculating loop and your agar plate. You're dipping that in your bacteria culture and spreading it evenly on an agar plate, storing it upside down at 25 degrees, etc. Now, once you've seeded the plate like this, you can then do the required practical. You can then test to see how different antibiotics are affecting the bacterial growth. So let's imagine you've prepared 2 uncontaminated cultures and you've seeded the agar plate. So this one is evenly covered with E. coli and this one with streptococcal bacteria. You're going to want to know which antibiotic works best to kill E. coli and which antibiotic works best to kill the streptococcal bacteria, because antibiotics are specific. They will work better on specific bacteria. So on each plate we have a paper disc that acts as a control. This is important because we want to prove that it is the antibiotic that is killing the bacteria and not simply the presence of the paper disc. So that would be the same size but without any antibiotics soaked in it. And the other 2 will be paper discs that are soaked in antibiotics. So for example, we could use 2 different types, antibiotic 1, antibiotic 2, and see which one is best at treating the bacteria. So you will set up the practical like this, then you will leave it in a warm place for several days, and then you'll come back and have a look at the results. So the results that you see might be something a little bit like this. For the E. coli plate here, the paper disc has got the E. coli growing right up to it. The whole region that's beige here is just representing the bacteria. So the control disc is worked and is not affecting the bacterial growth. The antibiotic 2 is clearly more effective because it has a larger area where there is no bacteria growing. This area we call the zone of inhibition. Means it's killed all the bacteria in that area and none is able to grow around that disc that's soaked in antibiotic 2, whereas antibiotic 1 is not very effective. On the other hand, for the streptococcal bacteria, you can see that antibiotic 1 is clearly the most effective. It has the larger zone of inhibition, whereas antibiotic 2 isn't as effective. Now, because these are circles, we might want to compare the areas of the zone of inhibition, which you do by measuring the radius. It might be easier to measure and more accurate to measure the diameter and then divide by 2. But regardless, we need the radius, and we can use pi r squared to measure the area of the zone of inhibition.